everybody. So let's start the webinar now. Okay, a very good afternoon, everybody. Welcome and thank you all for attending our second herbal webinar series 2022. We would like to give a warm welcome and thank you to Assistant Professor Dr. Pooja Shivanand for joining us today to present a lecture entitled Agarwood, Mystery, Make and Market. Just a few housekeeping rules before we begin. Please make sure to mute your microphones and keep your videos switched off during the talk. You may switch it back on during the Q&A session later on. May I now invite Dr. Nurul to give us a brief introduction on Dr. Pooja.
Thank you very much. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, everyone. A very good afternoon, Wednesday afternoon. So we normally have a Wednesday slot. This is the second herbal research webinar in 2022, organized by the UBD Herbal Research Group. So before Dr. Pooja present her some of her research findings, let me introduce her to you, sorry. Dr. Pooja Shivanand is an assistant professor and also program leader for the Environment and Life Sciences program at the Faculty of Science, UBD. And she has an engineering and... Oh, okay. Why is that? Can you see me? Can you see me? Right. Okay, she has an engineering and master's degree in biotechnology and also PhD in chemical engineering from India. She received DAAD research fellowship in Germany, and before joining UBD, she was a research scientist in biotech industry in India. UBD, she is the leading project principal investigators on various research topics through local and also international collaboration. Especially in the field of applied environmental microbiology, including microbial diversity in Brunei, mangrove endophytes, and their bioactive compound, disease prediction in food crop, as well as microbial induction of agarwood. Without further ado, let us welcome Dr. Pooja to share her webinar for this afternoon.
Good afternoon, Dr. Nurul. Thank you very much for the generous introduction. I hope I'm audible to you all. And also if you can see my slide.
Yes.
Okay, good. So let's today discuss about agarwood, the mystery, make and market. So as Dr. Nurul just mentioned, I am affiliated to Environmental and Life Sciences program in Faculty of Science, UBD. Before I begin the webinar, I would like to give you a brief background of where I come from. So if you look at this picture here, we are here in Brunei, yeah? And I come from India. This is New Delhi, that is the capital. So Brunei is about 4 degrees north of equator, and India is about 10 and over degrees north of equator. And I come from the west coast of India. This region I mention because it's quite similar to Brunei in a way, because it's also the coast and it is a biodiversity hotspot, the region called Western Ghats, okay? And in this region, when I was growing up, I was more accustomed to the perfume of a scented tree, which was sandalwood, which is another tree that grows in the region. And my state of Karnataka is called Gandhada Gudi, which is Gandha is sandal, and that state is the abode of sandalwood. Although I was accustomed to hearing the sound, the word of agar, I never gave it a thought of agarwood because sandalwood took dominance in the region that I grew up. However, growing up, some of the words that I could associate with agar was agarbatti. It's called incense, which is widely used in India. And also some of the ancient poems, some of the lines of some poems which talk about uh, thousands of years ago. They mention like agar, tagar, chandan, which means agar, turmeric, sandal, camphor, etc. So if you see this line, the text agar has primary importance. It's the first word that begins in the text. So that was the importance held then. And fortunately, it is after coming to Brunei that I could read about and research about my own words and associations growing up with this scent uh, after coming to Brunei. And I would also like to tell you that in India, this Agarwood or the incense is endemic to the northeastern part of India, this region here, mainly from the states of Assam and Nagaland and others, although now it is being grown in the other parts of India for commercial value. And there is a state called Tripura, the capital of which is Agartala. Okay, so if you look at this word, Agartala, okay, it's on the northeast of India. And the word agar, as we know, is the scent. I will describe it in some time. And tala is beneath. So the land beneath the scent of agar is the land of Agartala. So all these associations I could made sense to me after having embarked of research on agarwood in Brunei. So thanks to Brunei and this research for that. So I'll take you to my agenda. As I mentioned in the title, I'll be talking about mystery. What is this scent? Where does this scent come from? What is the tree that produces this? Okay, it's basically answering the question, what is agarwood? And then comes make, where we'll talk about how it is produced, how do we make it, how do you produce it, how do you manufacture it. And lastly, I will also walk you through the trades, because it's a commodity of very high volume, high value, being sold for thousands of dollars. A kg of agarwood can be sold for tens of hundreds of dollars. If you buy a few milliliters of oudh, which is the pure oil, 3 milliliters can easily cost you from say 200 to 500 Brunei dollars, for example. It's very expensive commodity. So we'll talk about the market and trade of agarwood. Okay. All right. So agarwood, the picture that you see here is the chips, the dried chips of the wood that has been harvested from a tree. What is that tree? We'll come to it later. For now, just know that it is a dark resinous wood that smells nice when it is lit. When you burn the wood, the fumes, the incense, that the smoke that comes off is having a very nice aroma. Okay, and this is what it looks like. And it's been mentioned in old Arabic and Persian texts as well, because the trade of agarwood is the market for agarwood is predominant in the Arab world, in the Middle Eastern part of the globe. Uh, and that is why I deliberately mentioned this text, because Goa is again on the coastal belt of west coast of India, where I come from, very near to my place. And this was mentioned that Sanskrit agaru was mentioned in a very early text. And you can see that it's called by different names in different places. Okay, it's called oudh generally, or gaharu in Malay, aguru in Kannada, my native language, or Sanskrit, jinko in Japan, Xinjiang. Uh, Pardon the pronunciation if that's wrong, but in China, in Vietnam, eaglewood in English. Okay, so it's called by different names, but essentially it means the same, a gift. Okay, a wood of very high quality, also sometimes called liquid gold, because of the value the commodity has. All right. So now this agar wood is formed from a tree. Okay, and that tree is called Aquilaria. It's also formed from some of the Gernop species, and not much, very few, 1 or 2 species, but Aquilaria 9 species, although about 21 to 23 species are known. It's largely produced by Aquilaria, and these belong to the family of Thymelaceae. All right, and this is, you can see the root here. It's Latin aquila, is meaning eagle. Probably because the texture, the color, a gray, brown, white mixture that the wood looks like, and that is how the word root was derived from, meaning eagle. So it's also sometimes referred to as eagle wood. And if you come to the species of Aquilaria, there are many. Majorly, people talk about Malacensis, which is found from the foothills of the Himalayas to the island of New Guinea, everywhere. Also from people talk about in Malaysia and in this Borneo, Southeast Asia region, the wood, Agarwood, coming from Malacensis. Now, since this wood is of so much high value and very expensive, it's there is rampant logging, okay, and trading in the wood. So most of the natural agarwood trees is lost. So they say about 20% or less is still remaining, and that is why this Aquilaria is put into something called CITES, a CITES database, which is based on international trade in endangered species of wild flora and fauna. Okay, so because it is endangered, and all the trade and export and import transactions, everything that happens legally is recorded on the CITES database. And Aquilaria is also listed on the IUCN, which is the International Union for Conservation of Nature. It is in the red list because IUCN, International Union for Conservation of Nature, lists the species as vulnerable, endangered, critically endangered, like Malacensis is moved from vulnerable to critically endangered. Okay. This is also listed in the IUCN red list. And in Brunei, the species that is recorded and is found is microcarpa and beccariana. Okay, species in Malaysia and surrounding regions could be Malacensis, Krasna, Filaria, Subintegra, etc. And in China, Sinensis. In India, it is Agalocha, which is synonym of Malacensis. But in Brunei, the majorly recorded species is microcarpa and beccariana. We checked this with the forestry forestry, and the leaves, based on the leaves of beccariana and microcarpa, these are different. Beccariana is a larger leaf compared to microcarpa, and the leaves were collected, the plant press was made, and it was referenced in Brunei National Herbarium Brun, and it was identified as microcarpa and beccariana species. Although in our conversations we heard that Malacensis is also found in Brunei, but sadly hasn't been recorded as far as I know it. And if you check or look at this phylogenetic tree, you see that Malacensis is very close to microcarpa. That is what an expert also told us, that it might confuse you with microcarpa, but the color of the wood of microcarpa is much darker, and that is how they differentiate it. But beccariana is a relatively far neighbor. So since it's not recorded, we identified and worked on beccariana and microcarpa. All right. So when we talk about agarwood, we know now that it is produced from a tree called Aquilaria. And if you can see this tree of Aquilaria, if you peel the bark, it is a very soft wood, white color inside, pale, and this wood is of not much value. All right. This brown thing that you see here, or the dark black or darker color, is something that produces the stress. And this formation of agarwood occurs by an intricately orchestrated stress response mechanism, which is in response to injury. Now that injury could be mechanical or insect bite, lightning jolt, animals, we don't know. There could be anything in nature. And in response to that injury, the tree secretes a defense mechanism, produces secondary metabolites, and that produces this resin. And in nature, it will happen. But in nature, it can take from 30, 50, 70 years. We don't know. Okay. And essentially, this is what is referred to as agarwood. So if I should tell you this healthy tree, white and nice, is of no value. The brown part, which is a response to stress, fetches billions of dollars. And that is how intriguing it is. And that is because of these oleo resins that is present in the heartwood. So I will show you a picture that we also came across. So this has been physically affected, this tree of Aquilaria in Banutan. Okay. And this white tree, white bark that you saw last here in the last slide, you see that it is very dark in color, brown, and this is what uh, is called the resin. It could be because of numerous external factors and could be because of microorganisms, fungi, bacteria, etc. And this essentially, they say that trees try to seal an external infection, which is comes under the concept of coded, which is compartmentalization of damage in trees. Okay, so a tree responds to an invasion, an external damage, basically, by developing compartments of decay. Why does that? It can prevent it from spreading to other regions. And this process creates a boundary to prevent the loss of normal tissues and to stop the spread of infection to healthy tissues. So this was also studied in Agarwood, where A. malacensis was shown to have an adaptation to wounding and fungal infection by developing a phloem boundary. So now in Aquilaria, the nice thing is, this is generally seen in many trees, in most of them. But in Aquilaria, the resins, the defense molecules that the tree produces is somehow aromatic. Although I must tell you that the etiology of these fungal infection, the diseases, is not a very well established study. It is still being studied, but a lot of concepts have been unraveled. So what has been understood so far is that in response to an external damage, the tree produces these resins, which fall under this category of chromones, chrominone derivatives, and volatile compounds. And these uh, are the compounds which impart aroma. So that could be, one of the experts told us, the Brunei oudh or the Brunei agarwood that is produced primarily smells phenolic. It's quite nice and very distinct from the other regions in the Borneo. So it also depends on the species of each tree. The infection and the defense and the resin composition varies from tree to tree. So it could be floral, sweet, aromatic, depending upon the ring structures. Okay. And that is the specialty of this Aquilaria trees. And there are some signature molecules in this category of sesquiterpenes and chromones. You can see that these are these benzene ring molecules, derivatives. And if you see these signatures in a GC profile or GCMS in a chromatogram, like agarofurans and agarospirals and uresmols, etc., ketones, aromatic compounds, you can be certain that this is an extract of oudh, of a high quality wood. And this is this is essentially, these resins are the secondary metabolites that the plant produces as a defense to external attack. So I hope this is okay. So this, so far we discussed about the introduction to agarwood. What is agarwood? Where does it derive its name from, what is the tree that makes agarwood, what are the trees found in Brunei, Malacensis, beccariana, and what is this mystery about that resin, which smells so great. If you can smell it sometime, you should really uh, give it a try. And then it is made up of, it is essentially a defense against external infection. So now we can move on to the second segment of my talk, which is how is this agar wood produced? Okay, this is the pale wood and this is the darker resin that forms. As I mentioned earlier, agar wood is formed naturally by animal attack, lightning, insects, ants, and microbes and other things over a period of time. It can range from 50, 70, 100 years. So now, because of its economic importance and high demanded product, illegal logging, deforestation has led to a loss of agar wood. And since it's a commodity that can become economic prosperity, it is tried to artificially induce agar wood in plantations. So these artificial induction, what they do some kind of a physical damage to the tree. So they either cut the bark or axe the tree a bit, so and leave it to heal. So in the process of healing, the tree can develop a resin, and that resin is scrapped off and either traded as wood chip or oil is extracted out of it. There's also biological infection where damage to the tree is done and microbes are introduced in the form of bacteria, fungi, and these fungi like mycorrhizas and bacteria, etc., they release certain enzymes which affects the tree, and the tree in turn uh, elicits the defense mechanism and produces the resin. It need not be just fungi and bacteria. In Assam, as I know, in India, they use the insects, Zurera conferta, so which naturally uh, is known to induce agar formation, and people are trying to use that in plantations to mimic the same response, so that the agar wood they get is uh, relatively as close to natural agar wood as possible. There's also chemical induction where they don't put anything, they just scrap the tree and introduce some chemicals. That if the chemicals range from sugars to some dilute acids, it's fine. But we've also come across reports that introduce iron nailing and toxic acids and all that. So we should also be careful where we are buying the agar wood from. Sometimes it may be very dark in color, but when you ignite it, you might just get a smoke instead of the incense. So these are there's a lot of tricks in the trade that is going on. So we were quite curious about this, and we had a discussion in our research group, and we thought natural agarwood is rare. If we could uh, try studying this mechanism of how agarwood is formed in response to biological attack, because we have previously isolated a lot of fungi and bacteria from the Brunei region. So we have embarked on a study in the past 2 to 3 years, in the past 3 years, called microbial induction of agarwood. And thankfully, this was funded by the Herbal Research Group and the IBER, Institute of Biodiversity and Environmental Research. And we did this study of inducing agarwood in the available Aquilaria trees in Brunei, in the Banutan Forest Reserve of the Forestry Department of Ministry of Primary Resources and Tourism. So our students, our collaborators, all of us, we in collaboration with the forestry people, we carried out some studies on Aquilaria for understanding what is really happening inside the tree. So this is a brief flow chart that I would like to share. So we selected some of the trees in the Banutan forest area in collaboration with forestry. And we used some of the fungal strains we already have in the department, because isolating fungus from Aquilaria and then inoculating it would be a long process. So we had isolated previously some of the fungi from Brunei locally. So we used that, made an inoculum out of it, essentially to grow the fungus in a liquid media. And put this fungus into the barks of agarwood, and then over a period of time, like say 1 month, 3 months, 6 months, 9 months, we wanted to see if there is any response, if the tree is at all stressed by this application of fungi. So some of the fungus we had already isolated, like trichoderma and cladosporium, aspergillus, penicillium, etc. So these strains, as I mentioned, these fungi were available in Brunei, available in our labs, and they are isolated from the forests of Brunei, like for example, from the Pulau Selirong mangrove ecosystems and tropical forest, mixed diplocarp forests of Brunei. And we had a catalog of all these strains from different regions. And 6 of these strains belonging to Aspergillus, Penicillium, Trichoderma, Cladosporium, we used to inject these agarwood trees and see if there is any response to infection. Now, we also wanted to understand, we had to have a control. Like, how can we say that it is just by a fungal attack, or is it just by a physical drilling of the stem, right? So we had two controls here. In one of the control, what we did is drilled the holes in the agar tree, but did not put anything. No media, no fungus, nothing, just leave it. And in another one, the drilled the hole and put the media of the fungus. Now, fungus is an organism that requires nutrients to grow, like you and I. So it's always kept in a sugar-rich media for the source of carbon and energy. So we prepare the media first. We sterilize it and we put the fungus. If you're from a microbial background, you would appreciate what I'm saying. Otherwise, you need to know that this fungus is grown in a sugar medium, which acts as food. So we just put the food, the media into the tree and not the fungus itself to see if there is any response. So I'll show you a couple of videos so that you could understand. Oops. Sorry, I'm taking a minute here. Okay, it's not running. Sorry about the glitch, just a moment. Okay, I'm back here. Sorry for that interruption. I would just like to show this video, how we drill the holes into the agar wood. Using this drilling machine, 8 mm drilling machine into the stem of the agar. All right. So the next one, once you drill the holes into it, we inoculate it by injection. And this project was carried out by my MSc student Yumni and her team of research students. So once the stem is drilled, we had this fungus inoculum, as I just said, and this fungus inoculum was injected into the stem. And we did it in different trials, okay? And in one of the samples, we also used just the media without any organism. Okay, it's just the sterile media, just put it to the hole of the tree to see if there is any response to the media without the fungus. Right. And after doing this, this was plugged by hammering a bamboo stopper. We had sterilized the stoppers and we plugged the hole. Essentially, when you drill, sometimes the water gets into it and all that. So in order to minimize such things, we plugged it. And then over a period of time, like say 1 month, 3 months, 6 months, we observed the reaction. So what is happening? Okay. Right. So I hope you understood the procedure. Sorry about that minor glitch. And then if at all there was a response and there was a dark resin formation, we wanted to check how big or how long is the infection and the area. And that is how we calculate if there was any significant induction done artificially. And we also try to collect these samples and distill uh, using a rudimentary Clevenger apparatus, hydrodistillation, take out the oil and study it. In general, if you even go to cottage industries, the soaking is done for like a long period of time, okay, a month's time, and the distillation process also happens weeks to 10 days altogether, and we do not have that kind of an apparatus, so it was a very rudimentary setup. Soaked it about for 2 weeks and then did some studies with that as well. And then we got the results of discoloration. Discoloration to the inoculation system, that treatments that we had made. And this finding we have now published in a journal on in Biodiversitas. And we could see this discoloration, the darker resin formation in our treatments. Just I am expanding it so you can see the discoloration. So around this is the white one, and as I said, the pith, the behind the pith of the tree, the peel of the tree, it is light color, which is not a resin. And if you look at it, then it has darkened. So the finding told us that there is agarwood formation. There is a response to uh, the artificial stress basically that we are giving the tree, and that was nice to see because it's working. To what extent? It's a very long term study. I mean, we can't do it for a year or two. It's a very long term study. Even I've contacted a lot of plantations in Brunei, in India, scientists in India and other regions as well, and they told us like even after artificial induction, they leave it for like 2 years, 2 to 3 years before they harvest the agarwood in order to get that aroma, so that it is very, very, very dense. Okay. And then we took out the oil. We need to get a GC profile of this or do some GCMS analysis, but we couldn't do it in our laboratory because of the lack of equipment. But we also built a collaboration with we are currently in the process of sending our samples because Aquilaria is listed in uh, CITES, right? Convention of International Trade in Endangered Species. It requires a lot of documentation to send the samples as well. However, with the help of our chemistry colleagues, thankfully, we were able to do a basic FTIR analysis and saw some signature molecules, the CH compounds, aromatic alkanes were detected, which are known to be associated with sweet fragrance of agarwood, as has been reported in the literature. So it was an indication, yes, there is a fragrant resin being produced. So this is the outcome of our study so far. So after this, the next plan was, as we were advised, that it would be nice to study, to inoculate these trees of agarwood from the microbes, that is bacteria and fungi that you find in agarwood trees. So we did a trial of that as well, which is still undergoing from a naturally infected agarwood tree in Brunei. Some of the samples were isolated, and we used it to isolate fungi and bacteria in the labs. And we could isolate a lot of bacteria and fungi in the labs. And we have cataloged them. And the fungus, please don't confuse with the fungus we used earlier. The fungus we used earlier for inducing agarwood were isolated from other regions, other forests in Brunei. These fungi that I'm showing you here, let me use the pointer, are the ones isolated from the agarwood, a natural agarwood that is found in Brunei. And we could isolate aspergillus, mucor, fusarium, trichosporon, etc. So trichosporon, mucor has rarely been reported. So this is a new understanding of the fungi in Brunei. So what we would plan to see is if this has any role of in producing a higher resin in future. But also there was an interesting finding. I remember I told you that we did have a control where we did not put any microbes, but just drilled it. So we could see a discoloration in that as well. So there was no fungal inoculant, just drilling the hole and leaving as such. And there was a discoloration, which indicates that it can also be induced by physical wounding without externally adding microbes. So this we have seen. But when we compare it with uh, adding fungi and not adding fungi, the area of the discoloration upon adding fungi is larger compared to this one. Uh, but I also discussed with other small scale business people in Agarwood, and they do say that it is inconsistent. That is the word they used. It is inconsistent at this stage to clearly say what kind of inoculum gives you the result. So a detailed study is yet to be made on this. And another factor we found is we used 6 different strains to inoculate the tree. There was no significant difference in this particular fungi giving a better yield. So most of it were able to elicit the agarwood formation. Trichoderma was much better as we studied. But what made a difference is the amount of time. Say, for example, we checked after 1 month, 3 months, 9 months, and 6 months and 9 months. The 6 months and 9 months area of discoloration was much better compared to the initial ones. So it shows that the time factor is important, that we have to leave it for a longer period of time. And physical injury, chemical application also shows agarwood formation, but application of microbes is also showing. So we need to discern or work on the formulation of what best to use for the local species in Brunei, because it varies from species to species. And we are going back some time and checking some of the trees there. I'll show you 2 pictures here. So once we scrap again further, we saw that some of the trees were healing and the other trees had these darker core. And we are not able to cut them further because we don't have permission to chop the trees, and we wouldn't want to do that at this stage when we don't have a concrete answer. But we peeled them superficially, and some of the trees have healed and some have a darker resin, and it's quite fascinating to understand why this is happening, and this process is going on. So that is about making or producing agarwood, which you should know. That in nature it happens naturally, but you can't wait. If you're in a business, maybe you will not want to wait for like 50-60 years, no, right? In the lifespan of a human. So they want to do it like 2 to 3 years, 6 months also in certain cases. And for that, they are using physical, chemical, and biological inoculation of agarwood. So now we move to the last segment of my webinar, which is the market. The market. So why essentially is agarwood important? So this agarwood, as I told you, is a very, very expensive commodity traded in billions of dollars across the world. And there's a huge market, especially in the Middle East, because uh, the Muslim population would prefer perfumes which are non-alcohol based. And that is also one of the reasons for such a high demand. And this is traded the world over in the form of wood chips, oil, incense, medicine, cosmetics, etc. Okay. The wood chips and oil of agarwood would look something like this. This is the chip of agarwood that has been harvested, and this is the oil that is made. What I was advised is not to just go by the color, because sometimes if you drill iron nail into the log of a tree, into the stem of a tree, it might give you a nice dark color, but it is all ferrous and gives out smoke and doesn't smell good. So it could be pale in color but also smell nice, so it's really hard to tell. And they say that it is a sinking wood, that if you put in water it sinks, but there is a lot of malpractice that people are using wax and other things, so we should be very careful while buying agar wood chips. Nevertheless, this is being used in perfumes, has a huge market in fragrance, in incenses, in cosmetics, and it's used as a fixative in some of the perfumes of large brands in Europe and America, and it is sent as a raw material there. And then also interesting is medicine. It is used in medicine, and this was beyond the scope of our current investigation. We didn't do any trials with it, but we have read a lot of literature, and there has been historic texts citing in Islamic texts and ancient Sanskrit scriptures in India, like Charaka Samahita in Gospels, etc., that it has been used for so many centuries for healing, in aromatherapy, in aiding digestive problems, etc. So it has been mentioned in Ayurveda and Unani and traditional Chinese medicines, in Arabic treatments, etc. And you can see a lot of literature on this as well. But of course, the dose makes the poison. So we need to keep in mind the concentration, the dosage, and that itself is an art when you are in perfumery. Right. So now I would like to show you some information on the trade of agarwood. As I mentioned in my earlier slides, that it is listed in the CITES database, and we have records from about 1995 to 2020 for the import. And Indonesia, import and export wise, makes up about 20% of the total volume worldwide. So a lot of trading is being reported from Indonesia. So way back in before Common Era, 1400s, it was mentioned as early as 1400 BCE that agarwood is described as a fragrant product in our Sanskrit text. So it's quite traditional. It's not last 50 to 100 years or so. It is quite ancient, I would say. Let me zoom this a bit. So how it is used for treatment, and I have some words also for you to look into, like Kannada is my mother tongue. I speak Kannada. It's contrary to the belief that everyone coming from India would speak Hindi, not so. So this is something that is called in my mother tongue, and where we have records of agarwood being used in healing. In China as well, the travelers found gaharu from Malaysia and called it Shenxiang, where the Chinese say that the incense doesn't sink in water, and probably that was the first test for agarwood authenticity used in traditional Chinese medicine as well. A lot of demand is also there for this in medicine. In Middle East as well, as reported in 800 CE in the Common Era, recently compared to these ones, that a combination of agar, which is aloes wood, and camphor, which is used as a scent for healing as well. And in the 1200 scriptures, I found this on the traffic that the Chinese custom official from Kwantung documented the supply of aloes wood, mainly supplied from the Malay Peninsula, from Sumatra, Borneo, Java, Cambodia, Hainan, etc. And they made a statement that Cambodian wood was quite superior as well. So I'm just citing this literature from what I've read to tell you the fact that it is not quite recent. It has been dated back to ages. It's ages old, the trade in agarwood and its use as a scent and also in traditional medicines. So Garcia de Orta wrote in his book Colloquies of Simple and Drugs of India that Chinese get their aloes wood from Malacca, Sumatra, and Ceylon. So it was a document that was sometime in 1564. So in India, again in Goa, which is a state near my place of Kumta in India. So it mentions that there were a lot of artifacts were made from agar wood, and there were mentions that Middle East connections has been there in the 16th century, 1600s, where trading was happening from the west coast of India, which is also called the Malabar coast, which I showed you in one of my earlier slides, to Persia and Arabia. And this is what I've got from CITES to show you how species like Aquilaria, we think that Aquilaria malacensis is most traded, but if you actually look at the data from CITES, you could see that we plotted this graph from the data that we got. From 2003 onwards, I think, Aquilaria filaria, the trade has been higher as compared to Aquilaria malacensis. I think I should also show you some of the trades live. From 1995, a lot of trade has been reported. Let me share the file. I hope you can see this file. Yeah.
Yes.
Okay, thank you. So if you see this file here, I've made this this graph from the data I sourced from CITES. And here you see, if I can zoom this, the dots in orange or brown, as you can see, what color. So this is a lot of export that is happening, and the dots in blue that you can see the world over are the imports that are happening. Okay, so if you see, let me zoom a bit. Indonesia, Malaysia, Thailand, Vietnam, Cambodia. So all of these, even Borneo, Malaysia, is a lot of export that is happening. I touched Brunei, and Brunei import is reported. Export is quite insignificant. This is all taken from the CITES. The data was plotted, and most of the other countries you would see in India, Middle East, UAE, etc. Most of them in US, etc. It is all in Saudi Arabia, North America. In Americas, it is all import, mostly import. And you can also see what is the kilogram of agar wood. This is all legal transactions that get documented on CITES. So most of the countries are importers, and some of the countries do re-exporting of agar wood. That is, they import agar wood and grade it and then re-export it to markets. I'll show you some other graphs that I plotted as well. Say, for example, this is the export by agarwood species. So here you can see what was the species of agarwood that has been traded widely. So for example, as I mentioned earlier, it is Aquilaria filaria, right? No, Aquilaria malacensis was earlier in the beginning. From 2003 onwards, filaria has caught up. And as I said earlier, Indonesia is the one that has been doing a lot of import and export in agarwood. So what is the species that is heavily traded? That is Aquilaria filaria, Aquilaria malacensis, Aquilaria filaria. Even Singapore is a higher exporter of agarwood with Aquilaria filaria and malacensis. Malaysia, most of it is documented as malacensis, and Thailand is Aquilaria krasna. Krasna is also a very nice species of agarwood, mainly traded from Thailand. So let's see some of the import which was plotted. This is again sourced, the data is sourced from CITES. The units are in kilograms. And you see these different species, malacensis, filaria, and malacensis, krasna, etc. That is, if you go down, you have other countries as well, China, Vietnam, India, Hong Kong, etc. But the amount reported on CITES is much, much less. Whereas Singapore, Indonesia, Malaysia, etc. Are known exporters and importers of agarwood. So this slide here, what I prepared was from the to know what is the commodity of agarwood that is traded largely, as you can see. So the chips are the most widely traded, followed by a powder of agar and whole logs as such. So chips coming from krasna, from malacensis, and undocumented species of Aquilaria, and also powder coming from filaria and krasna. So if you look at it, Aquilaria sinensis coming from China, malacensis, some also say malacensis, and Aquilaria krasna, and Aquilaria filaria. So these are the main species of agarwood that are traded, imported and exported compared to other 21 species of agarwood. Only 9 species are known to produce agarwood. And some of the species are also as synonyms. Aquilaria subintegra is also a popular species of agarwood. So next is the source. If you can see, what is the source of agarwood that is traded? Of course, wild will always be documented on CITES as far as I know. Then artificially propagated plant coming from krasna, filaria, malacensis, etc. Mainly from krasna. For the wild, filaria is the one leading. And also you have pre-convention, that is before the convention, unknown, etc. ceased. There is nothing, I think, yeah. I searched for some Brunei data, and not much of information is available though. It is mainly the exporting countries that export to Brunei. It is coming, I could only get for until 2017 for Brunei. So it's coming from Thailand and Vietnam. The export is coming from Thailand and Vietnam. 2014 from Thailand, China, yeah. So as reported on the CITES database. So far, majority of import to Brunei, it says it comes from sawnwood in the form of sawnwood from Thailand. Okay, let's see what about the oil export. I think I'm running short of time. I should rush here. The oil export, Malaysia is the leading one in oil. Although the species is not documented, it's Aquilaria species, and beccariana is traded. Beccariana is also found in Brunei along with microcarpa, and the oil that is gotten from this is traded from Malaysia, and from Vietnam it is krasna, Thailand, etc. What about the import of oil? Vietnam imports oil, Singapore imports oil, etc. Right. Okay, this was some of the data of import and export that I wanted to show you in my slides that is being carried out. Since it is an endangered species, it is always documented on CITES. That is the data we have. Coming to the last final parts, I'll briefly mention the business in Brunei. We also had the opportunity to interview and interact with some of the local business persons in Brunei, and they were very kind, I would say, and very generous to share with us the knowledge they have. We have Tuan Muhammad Salehin, a retired Air Force personnel, and he has a store in Center Point called Isan Oudh. And we visited his store, and he spent a lot of time with us and answered every questions that we had, and he also demonstrated to us in his own facility how the resin is formed and how he distills it. And he we can also visit the distillery. Because of these rising cases, we have the restrictions. Hopefully in the future we'll be able to visit, and we discussed and understood how business is being done in Brunei, and how they export oil and wood chips from Brunei. Another interview with Oud Al Aswad and with Haji Radwan, and he was called, he's being called the Agar man, as I said, in Brunei. And he was also very kind to answer all our questions on Oud, and he told us that uh, Brunei oud is very distinct, very nice and very different from other oud. It has that typical phenol scent, and a very nice a smell. And he has seen everything from the upstream of plantations and uh, maintaining, harvesting, distillation, and now he's into the downstreaming of the products, where he's into oil making, etc. And he has also kindly shared some of the uh, chips of agarwood that they process, the oil that they make, and also the oud baginda, which is the royal perfume that they. And because of these discussions, we had a very enlightening experience. Lastly, I would conclude here. Uh, in the interest of time, and I would like to tell all of you that it is a teamwork, and I would like to acknowledge the funding that we received from Herbal Research Group. So thanks to Dr. Norol, and co-funding from IBER, from Dr. Yati. I would also like to thank Dr. Rahayu here, though she is not directly involved, because it was in one of the discussions with her at Tropical Biodiversity Center. A discussion on agarwood was instigated, and that is how this project came into being, and she supported us. So thanks to her as well. And here you can see my collaborators. This is our project team. We have Dr. Hussain, who is the co-PI of the project, and we have members, Dr. Faiza and Dr. Yati. And since it is an endangered species also, there's a lot of paperwork and understanding with different departments in Brunei, and they simplify these things a lot. And I'm happy to be working. And my students, I must tell you that in this case, at least the students have kept me motivated in this project, because the location, the logistic challenges, and we don't have a very good plantations, very small trees, and uneven ridges and ranges where we have to walk. But I see these young students and girls especially, so how motivated they are. I had my RA Bazila, research assistant, and my MSc student Yumni, and they did it with like the experiments, all their experiments in the field with so much zeal and enthusiasm. I'm just showing them in work, because you can see how much hardships they put into the work. My student Zul and Mo is here, BSc student Hamima, okay, climbing the trees using the ladder. They did some experiments in nursery also, Fazatil, and my student Fadila, she's sawing a log of wood, which is found naturally, to see if there is any resin in it. So a lot of thanks to my students for keeping this going. From MPRT, I would also like to thank Mr. Zaidi, who answers a call each time we want to visit the Banutan Forest Reserve, Haji Dini, who has been very kind to accommodate us, explain us, take us on forest trails, and link us with the forestry people to help us. And thanks to all the forestry personnel from Ministry of Primary Resources and Tourism, for helping out with this understanding of Aquilaria and Agarwood in Brunei. So before I leave, I would like to leave you with a point to ponder on. Just think about it. We've talked about the mystery of agarwood, the mechanism, how it is produced, how it's traded, marketed and all. But at the end of the day, I would like to remind you that a healthy tree, well fed, well nourished, grows very well, no value. But a bruised tree sells for billions of dollars. Not just it's not the bruise that makes it so unique, it's how the tree defends itself, and how the tree encounters this infection, and in the process creates that very high value commodity that it is. So I would like to leave you with this statement. So what makes the perfume of your persona linger? So a food for thought, and thank you so much. That's the end of my presentation.
Wow, well done, Dr. Pooja. It's interesting, really interesting. Dear participant, guys, can we have a photo session first
Sure.
before we ask questions? Well done, Dr. Pooja. It's really interesting, and how you packed it into like a story. It's like mystery, make and market. I like you package how you package your presentation in such. I really understand how it works now. I think all of your team are here.
Yes,
You
yes,
want to
they are.
shout out to they're supporting you. I can see Dr. Yati, Dr. Hussein, they're all.
Yes, yes.
So
They are all my
guys,
team.
yes, if you can open up your camera, then we'll just for memory. We normally record and upload it for those who are unable to join us. Actually, a lot more people would like to join, but we limit our registration to 100 for the sake of. I would also like to thank Dr. Long, who sponsored this Zoom subscription. Hopefully, in the future, we'll negotiate on how to increase our quota more than 100. Dr. Long Meng, yeah, he's been very kind with our herbal research group.
I see.
So perhaps don't be shy. Maybe you still having your coffee. Don't go out yet, guys. Can we take photo? So turn on your camera, especially in support of Dr. Pooja. Where's the master student, the BSc? Dr. Hussein, where are you? All right, open your camera up. No one wants to open their camera. Can you hear me? Yes, yes. I'm audible. Dr. Hussein, do you want to put your camera on? Dr. Yati?
Yeah, I think there's a lot of shy people here.
Shy, very shy.
Shy student, yeah.
But they don't shy to climb the tree and do the sewing. Wow, amazing picture there. Nice teamwork. Yeah, I guess this is it, Dr. May. No one wants to open their camera.
Okay, we'll just take a group picture right now with whoever has their cameras on. I have with me here one full page of people with their cameras on, so I'll just take that. Okay, 3, 2, 1. Okay, another one on the next page. Hang on. I've got you here. 1, 2, 3. And the last one, with no one's camera on. Okay, 1, 2, 3. Okay.
Everyone is shy.
Yeah.
Right, it's question and answer. Don't go just yet. Anyone have a questions for Dr. Pooja? I have a question regarding your research. Yes, please. I saw that
Yes,
from
yes,
the chat.
yes, Dr. Nurul.
Yeah, please. Can you? I don't know what's that. Lemon 275.
Yeah. Lemon
Can you
275 has a question.
can you unmute yourself? I'd rather hear your voice than. Yeah, please don't be shy. Yeah. I have few questions regarding your research. Yes.
Yes.
She is still typing.
Sorry, I don't have any camera. Right. Okay,
Oh,
that's
okay.
fine. What is your question? Yeah. We'll just shout it out to Dr. Pooja. No worries. Any question?
She's still
Yeah.
typing.
Just I can still typing.
That's
One. Yeah, can I read it? Are there general characteristics to distinguish Aquilaria trees?
Yes, basically we distinguish Aquilaria trees based on their leaf structures. For example, the two species, I can say from what I have encountered in Brunei, like microcarpa and beccariana. So if you look at the structures uh, of the tree, of the leaves, they are very different. Beccariana is quite bigger and microcarpa is those small leaves. Okay, and we did an identification in consultation with Brun after we do the plant press and studying the leaves. Okay, also I think it comes from experience, because I was told that there's another question coming. I was told that it's sometimes difficult to differentiate between microcarpa and malacensis, but when they peel the wood, they see it from the color. So forestry people, I think, would be able to differentiate it quite well because of the experience of their association with trees.
Question number
Throughout your
2.
study, throughout your study, I'll read out the question. Throughout your study with Brunei forestry, where does these trees suitable to grow and distribute across Brunei? I've heard that with my discussion with forestry and also from the business people in Brunei, that there are a lot of trees, thousands and thousands of Agarwood trees in Brunei. But from my research on plantations in not in Brunei, plantations in general, what I've read is Aquilaria grows in a region well where there is no logging of water. If the water stands, then it is very detrimental to the roots. The roots become fragile, they rot, and the tree dies. So these trees grow well in a well-drained soil. Okay, so if it is a slopy area like a mountainous hilly regions, the trees of Aquilaria that grow are quite stronger than the ones that grow in other regions where there is a collection of water. And I was also told that if you prune the trees, they grow quite well as well. So if you want to grow Aquilaria trees, make sure that it's a very small investment. It is not a very fussy tree. It can grow as long as it gets water supply. I think for the first 1 to 3 years, you need to give it water and all. After it takes on its own. Hope I've answered that question. There's a next
That's a
one
good
come.
question. Regarding your visit to Brunei SME Agarwood, are there any specific classes of Agarwood and their price market? Does market price in Brunei similar to Malaysia and Indonesia?
Well, sorry, about the market price, I would not be the right person to talk about the market, but I would definitely recommend you to visit the stores of at least the people who I have encountered with, like Tuan Salehin, Ehsan Oudh at Center Point, first floor, and Oudh Al Aswad, Haji Radwan in Yayasan, and other small industries in Brunei, and they have different ranges of pricing. We saw their samples. We couldn't go to Oudh Al Aswad as yet because of the restrictions as of now we are avoiding. But we saw in Isan Oud that there are restrictions. There is pure oud oil that can cost up to 300 Brunei dollars and above. There are also dilutions, like for example, a bottle of oud as a base, which is mixed with other perfume like rose or any other scent, could be cheap, like $40, $30 for 3 ml. So it really depends.
Are you, madam, sharing a
Sharing
webinar slide? Yes.
Slides. Yes. I believe it's recorded, right, Dr. Nurul?
Yes,
Yes.
this is recorded. You can find it in our Herbal UBD website. So it's very important if you register. Oh, there's another question about the uses of agarwood. Is there any businesses or other uses of agarwood for medicinal purposes in Brunei? An interesting question.
Yes, it's a verbal conversations that I've had, and my understanding is it's quite used in the traditional medicines, traditional Chinese medicines, aromatherapy and all that. Since the medicine part was beyond the scope of our current investigations, we have not done any data analysis as such. But based on our conversations with people who deal with agarwood, we've heard that it's largely used for medicine as a stress relief, aroma therapeutic uses.
I've also read in one article in Frontiers, pharmacognostic studies on agarwood have shown anti-cancer. So yeah, that
Yes.
of interest, analgesic, anti-inflammatory, and antidepressant properties. But I believe these are not from the bark. Is it also from the bark?
It's mainly from the oil.
Oil.
Yes, mainly from the oil. But just for stress relief, even the incenses they use, like they burn the chip of wood, and the fragrance it releases is quite nice. But as I mentioned, the dose makes the poison, right? So we need to know how much to use and the applications and all that. Yeah.
Which are beyond this study. Yeah. There's another question from Dr. Suwari. Interesting thought. Thank you very much. Her questions: how many times do you need to inoculate the trees with fungi? One. Number two, how long? Where is it gone?
Okay, it's gone.
It's gone down. How long does it take to start seeing the discoloration? And what other kind induction can be used to induce agarwood production other than fungi?
Yes, I can say that. How many times you need to inoculate the tree? In our study, we did it once, but I have read that if it is a big tree, because our trees are very quite small, okay, and huge trees you can find which grow up to like 40 meters and all that. For those huge trees, I've read that you do multiple inoculations. You do inoculation once, and then after 6 months you do a booster dose. We are all familiar with booster doses these days. So that is also being done for Aquilaria. But a small tree, maybe one time inoculation. Going to your second question, how long did it take for us to see? As early as 1 month, we saw discoloration, because our trials were 1 month, 3 months, 6 months spaced. And as early as 1 month, we saw the discoloration. But generally, the thumb rule is this: the tree in plantation trees, example, they grow for like 8 to 10 years. When it is ready for inoculation, they inoculate it, and then they wait for another 2 years, 2 to 3 years, for it to develop into a denser resin before they can harvest it. What other kind of induction of agarwood can be produced other than fungi? As I mentioned, physical. So it depends. When people do business, they start with an end in mind. Okay, so what is the customer space, how much volume they have to produce? So generally they don't go for inoculating the entire tree. So what they do is they do peeling of the tree, and they apply like a homemade decoction, I would say, fruit enzymes and lemons and others, which gives the alkaline condition for the fungus and other bacteria to grow, and they apply it like paint okay, and they leave it, and it starts discoloration, and they harvest that peel. They don't sacrifice the tree, they harvest the peel, that segment, and then as need comes, they do physical inoculations. Okay, hope I've answered that question. But if it is
There's
like
so
for a very,
we're flooded with questions. We're flooded with
yes,
questions.
yes, it's
Muhammad
nice that
Aziz, thank you for the
Mohammed Azim.
insightful presentation. Dr. Pooja, given that agarwood tree species will become endangered, does artificial inoculation provide a way to sustainable farm agarwood and reduce the pressure from overexploitation?
I believe yes, yes, that's a good question. I believe yes. So what is necessary is sustainability, and for this, all the policies have to be there. Like, for example, the plantations have to be sustainable. So what they do is, for one tree they harvest, they plant 2 saplings of Aquilaria, something like that. Okay, and I believe rather than chopping off. And what they sometimes do with this illegal logging is, if you look at the Aquilaria tree, unless you are an expert, you wouldn't be able to tell whether it has resin inside or not. Okay, the resin is made inside the tree, not outside. So for that sake, they just chop off the tree, which is not nice, right? The tree dies, and you see that there is no resin inside. So in order to stop all that, if you have plantations, I think, in my opinion, that would be sustainable. But you should also work out, like, what is the marketplace, what is the processing unit, how much is the transportation required to go there. If there are many people working on this, an ecosystem will be developed, and there can be knowledge sharing and resource sharing and all that. So the policies have to come for that, to build business and all that. So a lot of factors will come into play, right?
Yes, we're flooded with questions. You can have a read.
Yes, is long-term plan of Herbal Research Group to develop the best inoculants to be used by local Agarwood. I would ask Dr. Nurul to answer this. I mean, is it a long-term plan by the Herbal Research Group? I can say you will get
Is that?
motivated students and a research team.
Yeah, I can foresee as also by sharing this knowledge gained by the student, it could also enhance the business industry in Brunei. I'm also one of the client because my husband loves the oud perfume. It's actually his friend,
I see, I see.
Ridwan is his friend, and they do
Fantastic.
sell.
Yeah.
They do sell with HM name, right? The
Yes,
Sultan.
Oud
Yeah,
Baginda. Yes. Yeah.
Baginda. So we bought that. It's actually expensive, and but I wonder whether is that like the market, is it fast selling or not? Because it's really quite expensive, small volume. Yeah, but I think in the long term, this is what we want, enhancing the student skill. This is a skill. This is a knowledge which not just developed in Brunei. And I'm amazed. Like Singapore is the country which exported most. Where do they plant it? They're such a small country as compared to Brunei, right? We have
Exactly.
more forest than them. Like, how come they're the highest exporter? So
yes,
yeah.
because they import it and they re-export it, because it's a trading hub. So a lot of demand from Singapore, because so a lot of re-exporting is going on. So that is another business strategy that Brunei
Or
could develop as well. Yeah,
I'm thinking they borrow the land of somewhere, Indonesia,
that is
maybe,
also there. Yeah,
right? But
invested
it's Singapore
plantations.
owned.
Yeah,
Yeah.
yeah, yeah.
Singapore
Invested.
owned. So I'm surprised the highest exporter like in the region. Indonesia, I'm not surprised. Yes. Saeed, after collecting the chips from the tree, will the tree die?
It depends how much you collect. If it's a small scale cottage industry kind of a setup, what Tuan Salehin told us, that he doesn't chop off the tree as such, but he just peels off a portion of the bark each time there is a necessity. So if it's a big company, then probably yes, they chop the tree off, because if you cut the lower portion, then the tree automatically dies. The peeling would save the tree, so you can keep it for a long time.
Again, from another, but the same participant earlier on. What
The
the main problem agarwood production in Brunei? Does inoculant Does the inoculant this SMEs using does not work?
We are yet to visit the SMEs, their distillation units, and to discuss on this. Probably the sourcing of agarwood in my, I'm believing, I'm guessing, okay, it could be probably the sourcing of wood. And of course, the inoculum, yes, definitely. When we had a discussion, I was told that there is no local inoculum, okay, and they import the inoculum from overseas, and most often it is quite expensive. You need to buy it again and again, and also you are not sure what are the contents of the inoculum. So is it really a live inoculum, or is it just a chemical, or is just a sugar syrup and all that? We do not know. So to answer your question, definitely it would be interesting to develop a local inoculum in Brunei. And when I had my discussions with like Haji Radwan, he is also more than happy to collaborate with our university and other places too, you know, and we can definitely work on formulations of local inoculum in Brunei.
Wow, I think guys, as we're flooded with questions, so a lot of interesting questions from you guys. Thank you very much. Thank you for
I have one
notifying me as
question.
well. Oh, Dr. Chan Chien Min, yes, carry
Dr.
on.
May is here.
May.
Yeah,
Yes.
Pooja, you
Yes.
said that your chemical analysis was you did your infrared with us, right?
Yeah.
Infrared. What about GC?
GC, we were told that there is no specific column.
Oh, I see.
Can we, if we are able to do GC here, we would be more than happy, Dr. Meng.
Depends on the availability of the standards, I think. as
Yeah,
well.
yeah. That's what the agarro spirals and all, they said. We checked with the department, so they were kind enough to let us do the IR. But for GC, we still haven't been able to. We are, that is why we are processing it to send it overseas, but a lot of scientist documentation and all that we need to clear.
It'd be good that we can do it here, isn't it?
Yes. So
Yeah,
probably
maybe
we can work on
something
that.
out, yeah.
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 Local collaboration would be very nice.
Sure.
Yeah.
Okay, thanks.
Thanks so much.
Right, thank you guys. Again, don't forget to fill in the feedback, what you think about our herbal webinar so far. Any suggestions, any if you have any collaborators that you would like to share, but it must be in the area of herbal, yeah. So thank you. One of the participants told me the camera setting does not allow for participant camera to be shown. So yeah,
Oh.
I thought everyone is shy, but yes, I doubt Bruneian are shy.
I'm glad they didn't doze off after my
Yeah, so
check.
they
Yeah.
tried to put the camera on but didn't show. Okay, never mind, other time then. Thank you very much.
Thank you
Dr.
so
Pooja,
much. Yeah.
especially thank you very much to make it
Hello.
Yes.
Hello.
I
Yes.
just want to, I don't know, point out because some of us already turned on our camera, but we only see ourselves and the panel, Dr. Pooja and
Can we
the
take a
host.
photograph again? Is that possible?
Okay, yeah, to make Dr. Pooja for her memory. Yeah, before we
Yes,
go.
if that's not too
Dr.
much
Min,
trouble.
what
On my
happened? The
screen, and I've
setting?
already taken the photographs.
Yeah.
So I
What
think I
happened
got
to the
everyone who turned on their cameras on the photo.
Okay, you can see.
Oh.
We cannot see.
Yeah, for some reason.
because-
I'm not sure.
Because I already checked with my friend and they said no, why nobody opened the camera, but the thing is we already opened it.
Ah,
Yeah, I don't know if the
see,
setting.
I
Then
opened
I
mine.
can't
Something to do with the setting because this is a new account.
Okay. We'll ask.
Be rest
Yeah.
assured though, I've already taken the screenshots of
Never
all the
mind.
participants who
Yeah.
have already turned on their camera.
Oh dear. Maybe I'm the
dear.
one who cannot see. I just see blank things. To be
Oh,
honest.
They get a pop-up message or something that it's yeah.
Never
doesn't work.
mind. But
Never mind.
importantly, we'll see you again next month. We already lined up our third herbal research webinar on 16 March. Join us with Dr. Mobin. Let me see. Normally my RA will email the same participant. Yeah. Okay, so that will be with the title will be Induction of Autophagy by Natural Product to Prevent Insulin Resistant and Hepatic Steatosis in Mouse Model by Dr. Mobin next month. Same again, normally we pick on a Wednesday afternoon because that's when we don't teach. Thank you. I do hope to see you guys again.
Thank you so much.
Thank you, Dr. Pooja.
Thanks a lot. Yeah.
Thank you, girls.
Thank you.
My 2RA who have been the background, as you said, we don't work alone. They are the one who've been emailing, making background, all these pretty things, posters, checking. Yeah, so it's also hard work. We also have to apply to university to get approval to conduct this event.
Right.
Thank you, Dr. Pooja, and thank you for joining people from Brunei, I believe, Brunei National Herbarium, Sungai
Yes,
Liang,
correct.
yes, yeah?
Yep.
I've invited them. Thank you, Dr. Pooja, for sharing insightful presentation. Yeah.
Thanks a lot.
Thank
Yes.
you. So do visit our Herbal website. If you just type Herbal UBD, it will appear on the first Google search. Yeah. Then from there you can see even the last month or coming months webinar if you don't have a chance to hear them. Yeah. On behalf of Forestry Department, I would like to congratulate Dr. Pooja and her team. Ooh.
Oh, thanks so much. Yeah, we're always very grateful to our forestry department, Mr. Zaidi, for always helping us. We call them up anytime and they're very, very helpful. Thank you so much.
Thank you, Tuan, for coming. Yeah. And if you have collaborators from forestry, if you like to give us an insight, some of this we also don't know, right?
Yeah.
Are being preserved in Brunei, what's important, what's not that important, because we're coming from health
Yes.
sciences. Yeah.
Yes,
Health
definitely.
science background. We also don't know what's on your part. Yeah.
Right.
So yeah, fill in the attendance form, fill in the feedback form. If you need an e-certificate, we'll email that to you. Right? E-certificate will be signed by our AVC, Dr. AB, Associate Professor Dr. AB.